Welcome everyone to today's webinar on using environmental data to inform water quality management in Buzzards Bay. My name is Paul Gannett. I'm an environmental product manager at Onset, and I'm very happy to say that we have with us today Rachel Jakuba with the Buzzards Bay Coalition, and she'll be giving most of the presentation today. This slide is for those that are having trouble with their audio. Here's a couple little pointers to look at. Hopefully everybody is able to hear me now. But if not, there's a couple places to check. And just a little bit of a tour of the control panel. There's this box here that says questions. You can open that up, and that's where you can enter in questions or feedback at any time during today's presentation. The webinar is going to last about 60 minutes, roughly 40 minutes to 45 minutes for the presentation itself, and that'll leave us a good 15 to 20 minutes for questions during the presentation. You can enter your questions in at any time during the presentation. And I also want to say that the presentation is being recorded, and we will make it available to all of you within a couple days after the webinar. So this is me. As I mentioned, I'm the product manager for environmental products. I've been with Onset now for 24 years, and I've had the pleasure of working with our engineers to develop our lines of hobo data loggers for outdoor and underwater applications. I live here on Cape Cod, Massachusetts, where we're surrounded by the ocean, and the ocean and the waters of Cape Cod are central to our economy and recreation. So protecting those waters is very important to me. And here's Rachel. She's science director with the Buzzards Bay Coalition since 2011. Rachel has overseen the coalition's science and monitoring activities, including a long-term water quality monitoring program that has collected data from over 200 locations around Buzzards Bay over the course of almost 30 years. In 2019, Rachel helped initiate a long-term salt marsh monitoring program around Buzzards Bay. Rachel also develops partnerships and secures funding for research and assesses technical projects in the watershed. Rachel received her PhD in chemical oceanography from the Massachusetts Institute of Technology, Woods Hole Oceanographic Institution Joint Program, where she studied nutrients and metals and their influence on algal dynamics, and her Bachelor's of Science is from the University of Georgia. At this point, I am actually going to turn control over to Rachel and leave you in her capable hands here. I'll pass control over. There you go, Rachel.
All right, thank you, Paul. Let me just get this in slideshow mode. Can everyone hear me okay? Hopefully.
Sound great.
Okay, excellent. All right, so thanks, Paul, for that introduction and for the opportunity to share our work with your audience today. So I'm going to take a couple minutes to just talk a little bit about our organization and the area that we do our work in. And then I'm going to switch to talking about how we use dissolved oxygen loggers to monitor coastal water quality. And then finally, I'll be talking about how we've used water level loggers as part of understanding nitrogen and phosphorus impacts of cranberry agriculture. Okay, so as mentioned, I'm the science director for the Buzzards Bay Coalition. So we're a nonprofit membership organization that was founded in 1987. We're supported by over 10,000 members, and our mission is we're dedicated to restoring, protecting, and the sustainable use and enjoyment of Buzzards Bay and its watershed. And we do that through working to improve the bay's ecosystem health through advocacy, research, conservation, and education. And anyone who is local, I would encourage you to check out our website. There's a lot of information in particular about ways to get outside, so to enjoy this beautiful slice of the earth that we live on our Discover Buzzards Bay website. All right, so to give you some orientation of where this work is conducted, so Buzzards Bay is located in southeastern Massachusetts. So on the map here, the sort of small map, you can see we're a little bit south of Boston and north of New York and Long Island. In terms of our watershed, so we have on our west is Narragansett Bay, and then up in the northeast is Cape Cod Bay, which we're connected to through the Cape Cod Canal, and then on the southeast we border Vineyard Sound and Nantucket Sound. And so the blue line on this map shows our watershed, and that area covers about 1,100 square kilometers. Our population, our year-round population, is around 260,000 people, although our summer population is quite a bit more than that. And the average depth of Buzzards Bay is relatively shallow, so it's 11 meters deep, or about 36 feet. And in terms of the impacts on Buzzards Bay, so excessive nitrogen inputs to Buzzards Bay have resulted in impairment of our water quality and living resources. And so I'm going to take a minute to talk about, if I can advance the slide, about what is nitrogen pollution. So nitrogen is an essential element for life, and it's generally the limiting nutrient in coastal ecosystems. And so when the amount of nitrogen coming into the system is in balance, you see water quality represented on the left half of this cartoon. So this is a healthy area where the nitrogen is in balance, and we have nice clear water, which allows dense eelgrass beds to flourish, abundant fish and shellfish, and it's also a place that's, you know, pleasant to get out in and enjoy. On the right side of this cartoon is an image of a nitrogen polluted system, where we have too much nitrogen coming in and we end up with cloudy and murky water where there's not sufficient light getting through for eelgrass to grow and thrive. We end up with low dissolved oxygen concentrations, and fish and shellfish disappear. And so to show you what that looks like in our area, here's some images of the effects of nitrogen pollution around Buzzards Bay. So in the upper left corner, we have macroalgae washing up in West Falmouth Harbor. And then in the bottom right and the upper left, these are both examples of rusty tide blooms. So on the bottom left, you can see the sort of reddish ribbons of water. And on the right, you can see it as this, you know, up close, it looks like this sort of golden, murky, rusty color in the water. And then in the extreme case on the bottom right is where we have a fish kill in the Acushnet River. So the coalition works hard to reduce nitrogen pollution and to protect water quality from having these impacts. And so I want a minute to describe the regulatory framework for protecting water quality in Massachusetts. And I'm just going to stop for a second and ask if everyone can hear. Uh-oh. Let's look in the chat. Or Paul, if you could unmute yourself, can we tell?
Yeah, we're getting a lot of questions. People are responding that they can hear you just fine.
Okay, great. Then I'm going to keep going.
Yep, sorry. I think you're good. Thanks,
okay.
Everyone.
Great, sorry. All right, so a quick primer on protecting water quality in Massachusetts. So the first piece of the puzzle is that the state of Massachusetts develops water quality standards that describe the desired water quality characteristics for different types of water bodies. Then every 2 years, the state of Massachusetts releases a 303D impaired waters list. So this is the 303D is the section of the Federal Clean Water Act that requires states to indicate where they have water bodies not meeting water quality standards. And then if a water body is classified as impaired, the state's required to develop a total maximum daily load. And once a total maximum daily load is in place, that's when pollution reductions and limitations are actually sort of enforceable. And really that's kind of, you know, where the rubber hits the road, shall we say. And in the state of, all right, sorry, oops. hold on, there we go. In the state of Massachusetts, nitrogen total maximum daily loads are developed on a site-specific basis. So what that means is there's not a set amount of nitrogen or a set nitrogen concentration in a water bodies in general that are targeted, and instead each embayment has to have the science and information developed for that specific location of how much nitrogen load can be handled and what concentration of nitrogen in the water body is appropriate. Okay, so for that first piece of the puzzle, so this is the Massachusetts Water Quality Standards. So almost all the waters in Buzzards Bay are classified as SA. And what that means is that these are areas that are expected to be excellent habitat for fish, other aquatic life and wildlife, and for primary and secondary contact recreation, and that these waters shall have excellent aesthetic value. So there are both quantitative standards and qualitative standards that, in order to measure whether these are excellent habitat for fish and appropriate for primary and secondary recreation. And in terms of the standards, so there's quantitative standards for dissolved oxygen, for temperature, and for pH. And then for the aesthetic values, so it's a qualitative standard, it's a subjective standard. In Buzzards Bay, we don't have an issue with heat pollution, and we haven't identified areas impacted by low pH, although there's pretty limited information on that. So really, when we're talking about trying to classify water as impaired or not in Buzzards Bay, what we're really talking about is whether or not the dissolved oxygen gets less than 6 milligrams per liter. And in terms of how that's determined, so every 2 years, the state develops this list of impaired water bodies, and with that effort for developing the list, they focus on monitoring a portion of the state's waters. So the state there's not a comprehensive or ongoing monitoring by the state of water quality for all waters. And that's where we come in. So the coalition's Buzzards Bay monitoring program was initiated in 1992 with two main goals. So first, to evaluate water quality and ecological health over time, and second, to educate citizens on the local water quality issues in their areas. And so the map on the right, the black dots are our monitoring stations. So we now have over 250 monitoring stations, and we monitor these with the help of about 140 citizen scientists. We've actually, for the last 3 years, we've had over 160 volunteers helping us with this monitoring. And the monitoring that they do, so about every 5 days, they measure dissolved oxygen, temperature, salinity, and water clarity using a Secchi disc. And that is from the end of May until late September. And then 4 times a year, we take a little bit of a deeper dive where we collect samples that are analyzed by the Marine Biological Lab for the full suite of nutrients, and also for algal pigments, so for chlorophyll and phaeophytin. Our methods are approved by the EPA and by the Massachusetts Department of Environmental Protection, or Mass DEP. In terms of our sampling scheme, so the dissolved oxygen, the sort of basic water quality measures there in the third bullet, those we measure between 6 and 9 a.m. And for the nutrients, we measure those in July and August on the last 3 hours of an outgoing tide. And the timing of both of those are designed to target when nitrogen impacts are likely to be most observable. So we're trying to catch we're worried about the worst case scenario, and so we're really targeting the worst case scenario with our monitoring. And the result of this program is that we have a really robust data set that's been rigorously maintained. Again, so next year will be our 30th year of monitoring water quality. And I wanted to talk for a minute about how we use our data. So we use our data internally to ground our advocacy efforts in sound science, and not just our advocacy efforts, also our land protection, and thinking about sort of all that we do. Having the information on where embayments are struggling more than others is really valuable for us about making decisions. We also provide our data freely to students and researchers. So if anyone out there is listening and wants a data set to play with, let me know. We also use the data for educating the community on Bay health. So this graph on the top right shows our Bay Health Index score. So this is an index score that we created that takes the varieties of information that we collect and calculates basically a eutrophication score from 0 to 100. It simplifies all those numbers into one that can be easily plotted and observed. And so this is West Falmouth Harbor, Snug Harbor. This is a harbor where you can see we've seen really significant water decline, and that's due to an increase in nitrogen loading from a wastewater treatment plant. And then this Bay Health Index, we use it as a simple communication tool. And so on the picture in the bottom right there, you can see that there's this little sign, and so we put out these signs all around the watershed where people are interacting with the water and where we have monitoring stations to let people know what the water quality is in their local area. Okay, and then what I'm going to be talking about today is how we provide the data to regulators that's really critical. And so this is both for using as information as wastewater treatment plant discharge permits are up for renewal or are initially being cited, and then also listing waters on the state 303D impaired waters list that I've already mentioned. I talked about how in the state of Massachusetts, our TMDLs, our nitrogen total maximum daily loads, are developed on a site-specific basis, and the Massachusetts Estuary Project developed, that was how those site-specific TMDLs were created, and it is the science that goes behind deciding what the threshold amount of loading for a given water body is. And then finally, to track water quality progress as changes are made to management. Okay, so the 303D list, that is the first step to get the ball rolling on protecting water quality. And this slide is just showing you a snapshot of 5 out of the many letters we have sent to the Massachusetts Department of Environmental Protection over the years to show, to provide comments on the impaired waters list and to provide data showing that, which water bodies really need to be on the list because we have this data to back up the low levels of dissolved oxygen. And the vast majority of water bodies that have been added to the 303D list are in our area, are the result of the Buzzards Bay Coalition data. Okay, so to have a look at that data, this is data from Quistit Harbor. On the left, there's the data from all the years that we've collected at station QH2. You can see, so that's the years on the x-axis, and we've got dissolved oxygen in milligrams per liter on the y-axis. The red line there is the 6 milligrams per liter line. That's our surface water quality standard, and you can see that we have plenty of values that fall below the 6 milligram per liter threshold. And then on the right, I took just 1 year of data. So this is data from 2019 to show you, a single season. And what you can see is that we really have very dense sampling compared with many other monitoring programs. So a lot of programs sample monthly or biweekly, but we're getting a much higher resolution in the summer months than that. Okay, and some of the advantages of our program are that it's low cost, so we have low cost supplies. We do have one staff member who oversees and manages the program, so that it's not no cost, but it's relatively low cost. And we're able to get a large spatial distribution. So with our 160 volunteers, and you saw all the dots on that map, we can really get very large spatial distribution all around the watershed. And it's also during the same time window. So we have set about 21 days over the course of the summer that we ask volunteers to go out from 6 to 9 a.m. So it's we're trying to get a snapshot many places in a narrow time window, which is really, we would not be able to do with staff. And then finally, there's also an important component of community engagement. So our citizen scientists really take a lot of pride in the work that they do. They enjoy getting out and doing this, although not always waking up early to do it, but they really, they feel a strong connection to their place. They talk to their neighbors and friends about what they're doing. And so for us, that's a really, it's a soft advantage, I would say, but it's a really valuable one for our mission. And then the limitations of this form of data collection are the frequency. So like I said, it's a lot greater density than many organizations in many places, but it's still only every, roughly 5 days, and we can miss events in between the sampling days. And then finally, I just note that the precision of the measurement is 0.5 milligram per liter. So our volunteers do modified Winkler titrations, and that is the precision that we're able to get. And so as the cost of water quality sensors have come down in price, and more water quality monitoring, more and more of the water quality monitoring community has begun using them, we've thought about their potential application to what we do. So instrumenting all the sites that we monitor would be cost prohibitive, and we don't want to lose the community engagement piece of our monitoring because we think that's really important. But there is some advantages to thinking about some higher resolution, higher precision data at some key sites. So this map on the right, the red dots are showing you what we call TMDL sentinel stations. So these are places where a total maximum daily load for nitrogen has been developed, and these specific dots are where those reports trigger a specific threshold for nitrogen concentration. So it's where there's focused monitoring to look at improvements over time. And in terms of, so we decided to pilot 3 loggers at three sites. So you can see we have 2 that are at a TMDL Sentinel site. So that's the Slocum's River on the west side here, SR4. And on the eastern bank of the bay, we have Quisset Harbor, or QH2. And then the final site we picked was Red Brook Harbor, or RB1. And you can see there's not a TMDL Sentinel site there, and that's because there is not a Massachusetts Estuaries Project report for that embayment. So this is a place where in the 2 where we already have a TMDL, we wanted to deploy there because we have critical information and a critical monitoring need. But for Red Brook Harbor, it's also important to start getting the information that we need to build that site-specific data set to be able to develop a TMDL for that embayment. And we first deployed these in 2019, so they've been deployed now for 2 years. Okay, so here's a look at our logger setup. So we had 3 loggers in in 2 of these setups, we had 3 loggers where we had a dissolved oxygen meter and a conductivity meter. In 2 of them, we had the pendant light temperature loggers as well. These were housed in a weighted fish trap, so it was weighted with a brick. The fish trap is very inexpensive. So again, as a nonprofit organization, we think carefully about how we spend our money. The DO logger is positioned so the loggers are held onto the fish trap with cable ties. It's positioned 30 centimeters above the bottom, and that is to mimic the monitoring that our volunteers do. We deployed these from docks. You can see the line there to tie it off at the surface on the dock, and we logged our data every 15 minutes, and we cleaned and downloaded the data every few weeks. Okay, so now to show you some of the data. So this is data from Quisset Harbor from last year, from over the whole season. So on the x-axis here, we have the dates, and then we have a number of y-axes. So we have the light intensity axis is the far left axis, and that pairs with the yellow data. Then our next y-axis is dissolved oxygen in milligrams per liter, and that pairs with the orange line. And then our right y-axis is for temperature in degrees Celsius, and that's the black line. And so if we look at the data, so let's start with temperature. And you can see that there's, there's wiggle in this line, there's daily variation, and there's also weekly variation. Although we do see, as we look over the season from early August to early November, we see declining temperature over time, as we would expect, with it really sort of dropping off once we get into October. And then if we looked at dissolved oxygen, so again, you can see there's daily fluctuations superimposed with these sort of larger weekly scale fluctuations. And if we look over the season, that in the sort of prime growing season, so in sort of August to kind of early September, We see really strong variations over time. So we're seeing sort of, relatively high peaks and relatively low lows. And that's reflective of, again, being in the prime growing season with a lot of phytoplankton busy doing their work. And then as we get later into the season, we see the variation in data we see a narrower range in dissolved oxygen concentrations observed, and they tend to be on the higher end. And then for the light intensity data, and this is new data that we haven't worked with before, so it's not a part of our existing monitoring program, and it was sort of neat to be able to add this little piece to what we're doing. But it's not something that it's the first time we deployed, and so we're still learning about how to use this data. And the light sensors, so these are deployed with the other things. So these are roughly 30 centimeters off the bottom. And so we're looking now at water column light. So what is filtering into the water column and through the water column. And again, we're seeing daily patterns that are superimposed by longer term patterns. And since the sensor is at depth, it's influenced by phytoplankton and particulates, and those influence what we're seeing. And to get more of a, so we have what's going on in the water column, both biologically and abiotically, and then there's also thinking about weather patterns and cloud cover over the surface. And with the sensor right at the bottom, we're just getting a slice of the information, and so it's a little bit hard at this point to interpret too much about what this data means. But if we zoom into, so that was the whole season. Now if we zoom into just 4 days worth of data, there's actually a really clear signal with the light intensity. So you can see, so again, we have the same y-axes and just a narrower x-axis that we're looking at. This is the same data set from Quisette Harbor. And you can see that as we start on a given day, as the light starts to filter into the bottom of the water column, we see the dissolved oxygen also starting to build as plants start to photosynthesize, and we see the temperature increasing as well. And then after we see our peak in light intensity, we end up with our peaks in dissolved oxygen and temperature, and they fall away over the course of the night and then build back up on the next day. So this is to show you an example of the different types of data that we collected, but now I'm going to focus really in on the dissolved oxygen, since that's the critical data for application to management. So this is data now from the other side of the bay, from the Slocum's River, from this summer. And again, we have the date on the x-axis, and on the y-axis we have dissolved oxygen in milligrams per liter. The gray dots are our logger data, and the orange dots are our volunteer data. And so if we compare these two, so for the citizen scientists, they collected 13 measurements over the summer. The logger collected in total almost 8,000 measurements over the course of the summer. But I'm comparing that, to compare it to the citizen scientists, I wanted to just look at the measurements in that 6 to 9 a.m. time window. And in that time window, it was just over 1,000 measurements that the logger recorded. And in terms of the 6 milligram per liter threshold for the state, so our volunteers observed 7 measurements less than 6 milligrams per liter, or about 30% of the overall number of measurements that they collected. The logger had 432 measurements less than 6 milligrams per liter, which was 41% of the measurements in that time window. So we're, with the data logger, we're capturing more of the low range. And if we look at the overall range observed in both places, so for the citizen scientists, they measure concentrations between 4.5 and 8.5 milligrams per liter, whereas the logger measured from down close to 0 up to 9.5 or 9.1 milligrams per liter. So on the high end, we're seeing similar concentrations, but the logger is really capturing some low events that the volunteers aren't. And you can see, it depends on when the volunteer sample was collected. So at the beginning of the season here, it happened that when the volunteers were out there, we're near a low point. But then as we get more into the season, some of those were missed. And so now I'm just going to zoom in from the previous graph to look at the volunteer data compared to the logger. And you can see on this graph, so again, we've got dissolved oxygen in milligrams per liter on the y-axis and date on the x, but we're zoomed into just a couple weeks now. And you can see that the volunteer data is really nicely tracking the logger data, or the logger data is tracking the volunteer data, whichever way you want to think about it. And if we compare all the data, so this graph on the right is the logger data on the x-axis and the volunteer data on the y-axis. Now we're looking at data not only from this station, but also from our other two stations and from both years. And what you can see is the data clusters around the 1-to-1 line, which is great, although there seems to be a little bit of a bias of the volunteer data to be higher than the logger data. And one of the advantages of the logger data, or of the logger, is that it stays on the bottom. So when a volunteer collects a sample, they have to bring the sample up to the surface. And we take great pains to limit any sort of oxygen introduction into the sample as it's collected. But it is not an infallible process. And so I think that there's the possibility that some of the slightly higher dissolved oxygen could reflect a little bit of oxygen entrainment in the sample, either sample collection or in the modified Winkler titration. Okay, so now I want to focus on identifying, so on this time period here in mid-August, which you can see is sort of between two volunteer sampling points, where the volunteers were near the low, not right at the low, but then there's this really massive low oxygen event that is missed. And so I want to zoom into that data. So this is again looking at the same data, the same station in the Slocum's River, and you can see we have these 3 days of really low oxygen, so less than 2 milligrams per liter. And these times of really low dissolved oxygen are very ecologically important. So it's these sort of extreme lows that can trigger things like it can drive fish kills, odors, species shifts. It's really where the concern. I mean, we're concerned whenever it falls low at all, but these extreme lows are really important data to capture and to understand that the system is periodically experiencing. And so then I wanted to layer this data with the other two stations to see how this compared other places. So this is data from Quisset Harbor, now in blue, and Slocum's River is still there in gray. And Quisset Harbor is a much less eutrophic station, and you can see that while there was sort of a little bit of a local low of dissolved oxygen in mid-August here, it's not the lowest that we see other at this station other times. And if we compare now the third station, so this is now Red Brook Harbor in orange, and this is another eutrophic site, and we can see that there is very low DO on these on 2 of the 3 days where we saw extreme lows at Slocum's River. And having this sort of fine scale information, it's a really interesting opportunity to compare when we see these extreme lows over limited time periods, thinking about what are the driving factors for those days where there was low oxygen. So being able to compare it to things like wind or cloud cover, or a variety of things that we can really start to dig into. And that level of resolution is not something we can do with our volunteer data. Okay, so to just summarize how we've used these loggers, so their application to managing water quality. So they provide high resolution information that we can use to identify impaired waters, to use as part of the scientific basis for developing total maximum daily loads for nitrogen, for tracking, as total maximum daily loads are in place or nutrient reductions are performed, seeing how the water quality responds, and then finally quantifying the sort of temporal variability and how it relates to other environmental variables, and also helps put the other types of monitoring we do in context. So understanding where the volunteer data fits in the overall, as a snapshot in the overall context of the data or what the environment experiences. So I think I will pause there if there are any questions for this portion of my talk before I keep going.
Yeah, Rachel, a few questions have come in, so maybe I'll give you those and you can address them. The first one was, what made the nitrogen rise so high in the waters you were monitoring? Do you have any sense for that?
Yeah, and I'm actually going to talk about nitrogen sources as in my next sort of segue into thinking about the next portion of research. In Buzzards Bay, what drives it mainly is wastewater. So we're around Buzzards Bay. We have primarily septic systems, and generally wastewater is the largest source to our embayments of nitrogen.
Oh, great. So we'll stay tuned for that.
Yep.
Let's see. Another question on, does the DO variability recorded by the logger, is it realistic?
I think so. I mean, I think that, that you can see the patterns of the DO data with time, and we, I mean, we can get very low. I mean, even so, none of the data I showed here, none of the sites here showed volunteer data as low as 2 milligrams per liter. But I have, there are other sites where I've collected such low values. So I think it is real.
Another question here on what tools did you use to manage and analyze the data?
So I work mainly with Excel, and which is, has its advantages and disadvantages. I know the program really well, which is why I tend to use it. I think that there's a lot of, as we start to use more and more continuous data, I need to look into some of the other tools to be able to process the data more efficiently. Like R is one that I'm really interested in learning, but thus far I've done everything in Excel. Well, and I should say actually the Onset HoboWare Pro. So we use that for exporting the data and for putting in calibration points at the beginning and end of our deployments.
Good, thanks. There's a couple questions here related to following of the sensors. What kind of procedures did you have to follow to deal with any biofouling?
Yeah, so we did put on the copper guards on our sensors. The fouling wasn't too bad, so we cleaned them about, like I said, about every 3 weeks. And often the fouling would be worse on the cage that on the fish cage as opposed to on the sensors themselves. But with that sort of regular cleaning, it wasn't to sort of take a stiff brush to them. It wasn't a major undertaking.
Good. Actually, you mentioned the fish cages. There was a question here. Somebody liked the fish cage setup. Did you find that there was much movement of the cages, and were they tied to some sort of dock, and how did you keep them in place?
Yeah, so in general, they stayed in place pretty well. So we would try and have the line taut enough that they would stay more or less upright. That said, we did have one cage for a portion of time end up on its side, and the DO instrument actually got some some mud kicked up onto it, and so we lost some data there where we just had to throw out that data. So in general, it worked pretty well. So we just tied it off to a pylon with a rope. But it is, they're having it. The reason we had them set vertically, the fish trap set vertically, was to gain that 30 centimeters off the bottom. But it did make it, somewhat less stable, although having the line generally worked pretty well to keep it upright.
Good. There's a few questions related to the loggers. I'll address those real quick, and then I'll let you get back to the presentation because I want to make sure you have time to get through that. There was a question on the cost of the loggers. The U.S. Price on the HoboDO loggers is $1,250. There's another question: do they have wipers? And we do not have a wiper accessory for the DO loggers at this time. Sometimes people There's sources. Zebra Tech, I think out of New Zealand, makes a wiper that can sometimes be fitted to the logger, but we don't sell those. And what is the lifetime of the sensors? And the sensors, they can be used for many years, but they have a replaceable sensor cap that has a 6-month cap life. So you need to replace it after 6 months. So if it's a seasonal deployment like this, you'll get the full season's worth of use out of it. If you're going to deploy it continuously, you'll have to replace the sensor caps every 6 months. So at this point, I do see one other question, but I think I'm going to hold off on that. Why don't you continue on at this point, Rachel?
All right, great. So now I'm going to switch gears and begin talking about, I've been talking about how we were tracking water quality impacts of nitrogen pollution. And now I'm going to switch gears to understanding and quantifying nitrogen sources. So this cartoon of nitrogen sources gets at is sort of a generic cartoon of a typical watershed where we have nitrogen getting into the atmosphere through things like fossil fuel combustion and a little bit of from farm animals that is then rained down on the water. We also have sources from wastewater treatment plants, from road runoff of impervious surfaces, from septic systems, which, as I mentioned, is a really big part of our watershed, and then also from crop and lawn fertilizers. And Buzzards Bay is a little bit unique because we have another source that's not typical for too many places, and that is cranberry bogs. And that's where I'm going to be spending the rest of my time talking about. So cranberry agriculture is an important to our region. So this map on the right, again, the blue line is the Buzzards Bay watershed. All the red area here represents cranberry bogs. And so we have a lot of cranberry bogs, particularly in the Wareham and Weweantic River subwatersheds. And I want to just, cranberry agricultivation is intimately associated with the water. And I just want, for people not from Southeastern Massachusetts, this is probably not a familiar topic. So I'm just going to take a minute to talk about growing cranberries. So So cranberries are grown in cultivated bogs. And so this picture on the right, so this is the bog bed or the bog surface. And those are generally surrounded by ditches. And the ditches have water control structures in them. And I'll show you in a minute in another image, you can see them. And the way water is controlled is generally to just put boards into the control structures that block the water or hold the water onto the bog, so they can either hold the water on or let it pass through. And these bogs are closely linked to natural streams and rivers and ponds, and they use that as reservoirs to pull in water for harvest floods. So the image on the bottom here, this is a sort of iconic image of a cranberry harvest. So the bogs are completely flooded and the berries are floated to the surface. And then the berries, so that's all this red, are the red cranberries, and then they're corralled and taken off for harvest. The bogs are also flooded for winter frost protection, and then water is used for irrigation, again, both for frost protection and for in the summer for growing the plants. And so the water to do all this is again pulled from natural sources, and it's also discharged to these same natural sources. So growing the cranberries, the connection between local waters is just very strong. So the motivation for this work was several-fold. So in sub-watersheds where there are dense cranberry bogs, the cranberry agriculture can be a significant source of nitrogen to coastal waters. Though, again, wastewater is usually the largest source in our area. But these pie graphs on the right show the balance of sources. The sort of tan color is our wastewater, and these are two subwatersheds of the Wareham River. And then the pink color is the cranberry agriculture. And you can see, so where we have dense cranberry bogs, it can be an important source of nitrogen to coastal waters. And both the Wareham and Weweantic River, where we have our dense cranberry agriculture, they're both impaired waters that have seen, once you get down to the coastal portion, have seen loss of eelgrass as a result of nitrogen pollution. In terms of how we get the number that is associated with these pink sections of the pie chart. So there's really not as much data. There's limited data on how much nitrogen and phosphorus cranberries export to the system. And again, so cranberry agriculture, it's a relatively limited geographic thing, and so it's not, there's not a myriad of studies around there about it. So this was, and because once you get to developing a total maximum daily load, this is where the sort of, the rubber meets the road and actual reductions are required and people have to pay money to make changes. And so there's a lot of interest and controversy around how these numbers are developed. So we wanted to, number one, improve the data certainty around these numbers, and number two, also by getting information about how the nitrogen and phosphorus is coming off the bogs, have the potential to suggest or propose reduced nitrogen and phosphorus discharges. Okay, so the way we did that was we wanted to quantify nutrient inputs and outputs for 1 year in 3 representative bogs. And by a representative bog, what I mean is a wetland style bog with typical fertilizer management. And there's a couple other types of bogs, but the wetland style bogs are the dominant source, the dominant type of bog in the watershed, and that's why we focus there. And if you think about what the inputs and outputs are, so this cartoon on the bottom shows inputs including fertilizer onto the plants, again, atmospheric deposition, and water, surface water carrying nutrients. And then our outputs are the surface water going back out, the harvest of the berries and the leaves that come off during harvest. And then you see there's sort of question marks for the groundwater discharge or recharge. And that's because, depending on where a bog sits in the watershed, it can be either a source of groundwater input or output. And we had done some previous research where we had tried to do, or we did do some grab sampling to look at, the inputs and outputs. But what we really, in doing that research, what it demonstrated was that without having really good information about the volumes of water moving, that introduced a large source of uncertainty into the overall amount, understanding of the overall amount of nitrogen that was going off of these bogs. Okay, so how we accomplish this? So we, in order to get a really good information, so we wanted to both quantify the water volume and then quantify the nutrient concentrations and pair those 2 sets of measurements to calculate nutrient fluxes. And so in terms of water volume, so we measured precipitation, surface water inputs and outputs, and that's, I'll talk a little bit more about how we did that. We estimated evapotranspiration from the plants, and we calculated the groundwater exchanges by difference. And then for quantifying the nutrient concentrations, so we had automated water sampling for chemistry during these different phases of water management or bog management. So during the harvest floods, during the winter floods, during what we call base flows or the sort of interim times, and during summer rains. And in terms of what we measured, so we looked at forms of nitrogen, so both inorganic and organic, and then both, again, inorganic and organic forms of phosphorus. Okay, and so the instrumentation that we use, so for gauging the water volumes, so we gauge the inlets and the outlets to calculate water flows. And so the middle image on the right here shows a flume at the outlet of one of our sites where we had the water level logger that allowed us to calculate the flow coming through that flume. We also had water level loggers in the ditches, so we could compare the water level on the bog with the water coming out into through the outlets. And then to understand how much water was being put on by frost and irrigation, the water pumps were gauged so that we could get that information. And for the water chemistry, we used ISCO automated samplers that are up here on the right here. And this is one of the water control structures. And actually you can see those are boards that have been pulled out of here. And we also collected water samples from groundwater wells up and down gradient of the bogs. Okay, so the goal of collecting all this data was to be able to have information about the water balance for different phases of management, and then also the nitrogen and phosphorus inputs for these different phases of management over the course of a year. And having that information allowed us to be able to identify periods of high nutrient export that would make sense to target for reductions in nitrogen and phosphorus outputs. Okay, so this is an example of the bog water level results. So we've got stage in centimeters on the left y-axis and discharge in liters per second on the right y-axis. And again, we've got our year from late 2015 to late 2016 along the bottom x-axis. And you can see that we have this 0 for stage. So this 0 line is where the surface of the bog bed is. And so then these, where the stage is below the bog bed, that is where we have water in the ditches, but not as high as the surface of the bog. And if we look over the course of the year, you can see there's only a few cases where we have water over the top of the bog. And that is here, our harvest flood in late October, early November. And then there's a series of a couple different winter frost protection floods between January and April. And associated with these high peaks in stage, we have, once the stage, in order for the stage to fall, we have the discharge coming out of the outlets, and we see these big discharge points associated with those floods. And in the interim, where we have little bits and pieces of increases in stage and discharge, there is where we think we're seeing either storms or rainstorms or little bits of irrigation that are that make it into the ditch, but not all the way up onto the bog. Okay, and so what we get from that information for the 3 different bogs, so this is data, these ranges are from our 3 different study sites. So we had precipitation as a water input of about 1,000 millimeters per year. Our harvest floods here, we have relatively small variability between the 3 sites. So between sort of 400 and 650 millimeters per year. The winter floods we have a more variable amount of water. So depending on the exact setting of the bog, some bogs flooded more than others, depending on the farmer's preferences. And then, so for both the irrigation for frost and for summer, again, we're looking at a relatively narrow range between the 3 bogs. Where we see a really huge range is in our groundwater. So again, depending on where a bog sits in the overall, its hydrographic setting makes a big difference in terms of whether it's a source, whether groundwater comes into the bog or leaves the bog. And we had one bog in particular where there was a lot of groundwater coming into the bog. If we look at the outputs for the water budget, so we had a calculated output of evapotranspiration. Then we have for the harvest floods, we have between 300 and 700 millimeters per year. Relatively low variability again, but higher variability when we look at the winter floods. And then we have what I'm calling our non-flood base flow. So this is the times of year when there's not active management of water on or off of those bogs, and that is our biggest source, or our biggest piece of the puzzle, and it's also the largest variability. And then we have rainstorms, which were relatively low in the year that we looked at, and groundwater, which again there was one bog where there was actually an output to groundwater. And so the main messages here are that the water use is similar in our harvest floods and irrigation. There's more variability when we look at winter floods. The hydrogeographical setting varies and is very important in terms of the water budget. And most of our water is leaving the bogs in non-flood base flows. So now to look at the concentration data that we paired with the flow data. So I'm actually going to start with the image on the right. So this is particulate organic nitrogen in milligrams per liter on the y-axis, and hours in the x-axis. And we have the gray dots are water that is coming into a harvest flood, the supply of water, and then the open diamonds are the water coming off of the flood. And this is time from the start of the flood. And you can see that the water in has low and consistent particulate organic nitrogen. We see a peak in the first part of the flood. So in the first sort of couple days, there's a high output of particulate organic nitrogen. So this is when there's a lot of movement of water fast, and so it can stir up the water and carry some of those particulates. Whereas the nitrate here on this left panel, we see not much movement of nitrate in the first couple days, and it's only as most of the water has moved off the bog that we start to see the nitrate concentrations coming up. Although if you look at the scales, it's much lower concentrations of nitrate than particulate organic nitrogen. And we think what's happening here is that once the water is below the surface of the bog, then we have the water in contact with the sediments that are, where a lot of the plants are, and it's sort of squeezing out some of this high nitrogen water once we get down to that portion of the flood. And if we look at the phosphorus release, so here we also have an early release of phosphate. So it's in that first, really 12 hours of the flood that we're seeing most of the phosphate coming off. And again, so the what's coming off is in the open circles, whereas what was going on is the gray circles, which is very consistent. All right, so when we combine the concentration data and the water data, we use that to get our annual fluxes. And so there were 2 bogs that were small net sinks of nitrogen. There was 1 bog that was a large source of nitrogen, and all of the bogs were small sources of phosphorus. And in terms of the importance of different water sources, so the percent of those fluvial outputs in the different water sources, so for nitrogen and phosphorus, again, it's the non-flood periods that were the dominant source of nitrogen. If we wanted to target management, the non-flood periods are where we would want to target, because that's where the majority of the nitrogen is coming out. Although we do have some coming out, nitrogen coming out in harvest floods, more phosphorus in the harvest flood. And the rainstorms here, we really saw very little nitrogen and phosphorus, although I have an asterisk there because it was a drought year that we were looking at. So in a year where there were more intense storms, it could be higher. So the applications to management for this work. So where we would want to address nitrogen phosphorus is really in the continuous water output. So that's where we have water that's high in inorganic nitrogen coming out during the growing season. And there's a potential to discharge to streams with high nitrogen uptake in complex channels. So types of actions that could be taken are really working to make sure that we naturalize downstream areas so that there is the ability for high nitrogen uptake by the vegetation, or putting water into through like holding ponds to give it a chance for uptake in the ponded area. The most available forms of nitrogen are high late during harvest flood release when water flux is low, but for phosphorus, it's the opposite. So it's at the start of the flood discharge. So trying to address the nitrogen and phosphorus that comes out during a harvest flood would be difficult with one strategy. And then the nitrogen and phosphorus that's released in winter floods is less important, and the storms is sort of a TBD because we were measuring during a drought year. So I just want to wrap up by acknowledging my colleagues at the Buzzards Bay Coalition and the Woodwell Climate Research Center. Chris Neal led the Cranberry research along with Casey Kennedy. We had great cooperation from the Gilmore Cranberry Company and help from the University of Massachusetts Cranberry Station and Rich McCorney at the MBL. So I will wrap up there and happy to take any questions.
It'd help if I unmuted myself. We have a couple questions here, and I know we're getting close to the end, so I'm going to take a couple. I'll take this first question, then maybe we can advance to the contact slide so that people that need to follow up with us will have that information. But if you've got time, Rachel, we can hold on even longer to answer other questions.
Yep.
So yeah, the first question I have here is how was discharge measured, directly or calculated by using stage and level, or stage level? I'm sorry.
Yeah. So by calculating. So we had with the flumes, we had the measurements of the flumes and then the water level in order to calculate the discharge.
Yep, that makes sense. The next question I have is actually back to your the prior segment. The question was, how deep was the 30 centimeters off the bottom? In other words, what was the water level above that?
Water depth. Yeah.
Yeah.
So they're tidal sites. They're generally, so I think Quistit Harbor and Red Brook Harbor are both about 4 meters deep overall, and Slocum's River I think is a bit shallower, but I'm not positive about there. But they're relatively shallow waters where we were deploying them.
So let's see. We had a question here. I'll take this one, which is how to best collect conductance data in estuaries where conductance is between the low range and the high range. That's obviously, this question comes from somebody who's been using our logger. We have 2 ranges on our conductivity loggers. And I guess that wasn't really part of this presentation, so maybe it's a little bit off topic, but I'll address it real quickly, which is you can log both the high range and the low range at the same time. And then depending upon where the data is, or where the readings are, you use the data from the appropriate range where it's going to give you the best accuracy. So actually, it looks like we've covered, unless I've missed some in here. And if there's any, we'll kind of go through them again. Oh, I see a couple more. Question about did we use cellular or Wi-Fi data gathering methods, or whether you did. I think these were all manually offloaded. Is that correct?
Right, we didn't. Yeah, so because we're trying to get the measurements at depth. So again, our interest is really the worst case scenario. So trying to focus on data at depth, that's where we have, it is more difficult to have real-time connections in terms of the hardware. And also, I mean, from when I've looked at the idea of having the hardware connected, it's not an insignificant cost in general to add that, and that has been part of our decision to just offload the data every few weeks.
Mm-hmm. Makes sense. Well, I think we're a little bit over, but hopefully it looks like most people stayed on. So at this point, I just want to say thank you, Rachel, for sharing your insights with us today. I found it really interesting, and I'm sure everyone else did as well. And I want to thank all of you that attended today. I hope it was you found it worthwhile, and that you will attend our webinars in the future. You can see our contact information on this slide. So if you have any questions, certainly use our website. It's a great resource with a lot of tutorials, webinars, just a wealth of information. All of our product specifications and pricing is there. You can see Rachel's contact information there as well if you want to follow up with her directly. And she said that would be okay. And feel free to give us a call or send us an email at Onset at any time. So thank you again for everyone, to everyone for your attendance today.